All right. Next, we have Brian Searle. Brian is one of the founders of Proteome Software and is currently at the Institute for Systems Biology in Seattle, Washington. And today he is going to present a talk entitled Building Libraries for DIA. So with that, I will pass it over to him.
Hi, Jacob. Thanks again for the introduction. It's too bad that we can't do this in person, but I'm really glad that we can still do some sort of user group style system for you or a presentation for you. The sort of stuff that I wanted to talk about here was kind of more general purpose. How do you actually start to use some of these libraries that Nick was talking about developing and that Mac was talking about using at the end of his session, and how to actually build these libraries and then what they are most beneficial for. So before I begin, as Jacob said, I am a founder of Proteome Software. I also have an academic or a research laboratory at the Institute for Systems Biology, and the funding sources for this are from both sources. So with that, I'd like to talk about library free detection and also sort of the limitations of that style of work. And then second, about building DDA-based libraries and how to best build these libraries and how to best use them. And then second, discussing more about these predicted spectrum libraries, specifically with PROSIT, and how they can be used for DIA and what their benefits and drawbacks are. And then finally close out with some new work on DIA-only libraries. So just to kind of cover some of the basics of DIA again, if you haven't seen this before, I'm just showing a precursor map on the left side, separated by retention time and several precursor MCs. So each of these is a different peptide. Each of these curves is a different peptide. And with DIA, you take a stripe or a section of that precursor mass space and you co-fragment all the peptides simultaneously into the same MS2 spectra. And this gives you fragmentation traces that represent each of those individual peptides. While any of these individual MS2s are often extremely complicated because they have co-fragmented peptides in them simultaneously, if you start to think about time aligning these traces, you can deconvolute some of this information from itself. It's essentially a mechanism to multiplex peptide measurements where you're collecting multiple peptides in the same spectra. So given that, there's a lot of, I think, interest in why DIA is so useful and why people are interested in it. And I think that it gets around a lot of the concerns of doing DDA analysis. And so Max sort of touched on some of these a little bit, but I kind of just wanted to stress what these are and where they come from. So for example, there are many cases in which you'll find situations where you get an early DDA trigger based off of either a shoulder of that peak or another, or actually just another co-eluting analyte. So for example, this is a peptide where there's actually two analytes of exactly the same precursor mass, but only one of them is the peptide of interest. And in this case, you would stick the precursor mass on an exclusion list and never sample it the second time. Here we're able to get really nice chromatographic peaks for each of the different fragment ions for a peptide like this. So this would indicate a type of peptide that would be missing. Another aspect of this is that these precursor traces also can oftentimes be interfered with. So one of the disadvantages of DDA with precursor ion integration is that you really have three different numbers. You've got a precursor, a monoisotopic peak, a +1, a +2 peak. And if any of these are interfered with, well, then that number essentially, or the quantity for that number is in question. And there's not really much you can do to recover from that. Here, if we see interference at the fragment ion level, it's fairly easy for us to say that one of these fragment ions, in this case, there are 10 different or 12 different fragment ions that are possible to be used for quantitation. If one of them is interfered with, then it's very easy to kick that out and use the other 9. to do quantitation with. And then finally, there are many precursors or many peptides that actually don't produce any signal as precursors. And so, for example, this is a peptide where we see really nice fragmentation patterns where they coalute and indicate the shape of a peptide, but there's actually no signal that indicates that this precursor is present. And this happens actually a lot more frequently than we would think. In fact, all of these three types of things happen a lot more frequently than we think. I took one data set here and then sorted it based on alphabetical sequence, and then found all of these threes within the top 30 peptide sequences. So these are all very common situations for us to see peptide fragmentation happening. And it's, I think, worth thinking about why this is going on. So for example, if you were trying to think of analysis in orbit trap, if you were doing normal DDA, you would take a bunch of ions in your precursor scan. You would put all of these ions, pass them through the quadrupole, and then put them into an HCD cell to accumulate so that you would get enough ions to make your measurement. Typically, we accumulate such that you can look at, say, 3E6 ions on a QE or 4E5 ions on a Lumos or a fusion instrument, and then you would measure them in an orbit trap, and then you would see their intensities in the readout. So what happens, for example, if one of those ions, for example, is a little bit lower in population than the others? If the population in the HCD cell is such that most of those precursors are from one or two species, they can actually swamp out all of the signal from any other peptide. And so, for example, that orange trace will just never get seen because it never makes it, or not enough of those ions make it into the HCD cell to fit within that 3/6 limitation. So DIA takes advantage of this, as well as the boxcar method that was developed by the Matthias Mann group. And both of these strategies essentially use the quadrupole to section out peptides so that you're only storing and you're only accumulating certain groups of peptides in the HCD cell. And because of that, you always get some nice measurement. With boxcar, the strategy is essentially just to collect very accurate precursor signals and use that for detection and quantitation. DIA is essentially the same sort of a strategy where you're using the quadrupole as a filter to accumulate different precursor ranges. The only difference is that you get fragment ions that are associated with that peptide, and you can use those fragment ions for quantification. So when you're trying to detect peptides with a spectrum library, for example, or other sort of technique, you'll end up having to extract out a bunch of fragment ions. So this is, for example, a set of signals that have been extracted out from a specific peptide in a DIA file where I was interested in trying to measure this peptide. And so it's really difficult to tell which of these signals indicates the actual presence of that peptide. And here I've indicated where it is. It's actually in a relatively low abundant region of the chromatogram. And so here there are two different sets of signals that might indicate the presence of a peptide, or a few different signals. And it's really difficult actually to try to figure out which of these signals is the true signal. So for example, there's that peak in the middle at 64 minutes. There's that peak over to the left at 63 minutes and the one over to the right at 65.5. And it's really difficult to determine which of these peak groups actually represents the peptide. The way we typically do that, so for example, in this case, it's the peak group on the right, is by using these library spectra to be able to interpret the relative abundance of those fragment ions. So as opposed to normal DDA analysis where you would use a search engine that doesn't necessarily know about prior information, here we really need to rely on the presence of prior information to be able to be confident in these types of detections. So for example, if you look at the actual apex of the peak, the mass spec signal there is actually a composite of many different peptides simultaneously. But if you look at the most abundant peaks, you actually see the same fragmentation pattern throughout. And so that is how you derive confidence in this peptide detection. So there are multiple ways of doing peptide detection or peptide analysis here without using anything or any type of library, just using a FASTA database. So for example, this is a strategy called PECAN, which is peptide-centric analysis, where you basically take that same fragment ion spectrum or a chromatogram set, and then you score each of those different mass spectrum signals, each of these different time points. You would actually score each of those scans, and at each of those different scans, after deriving a score, you would see that there would be a potential peak in those score distributions. If you were to zoom in around that peak in those score distributions, that would be the way in which you would determine that this peptide was present, and how confident you were in that score set. So for example, Scaffold DIA has a method to do this using a program called Excordia, which essentially is a version of Sequest, or the Sequest scoring engine, but restructured for DIA analysis. So these types of methods can help you detect peptides without ever having seen them before, but they tend to not be as sensitive because they don't know anything about what retention time you should be looking for and what fragmentation patterns you should be looking for. Another way to kind of do this is using a method called DIA-Umpire, developed by the Nisvisky lab in 2015, and has been recently re-implemented as direct DIA and Spectronaut and that sort of thing. But these types of methods essentially try to extract out co-eluting fragment ions that are associated with a specific peptide and make a pseudo-spectrum. And the way they do that is by looking at a monoisotopic peak and in a precursor signal, and then also look at fragment ions that are associated that seem to co-elute with them. They'll find those co-eluting precursors, map them together, and essentially derive a pseudo-spectrum that can be fed into a normal DDA pipeline. These types of methods actually are starting to work fairly well and seem to be reasonably robust. But there is some disadvantage in them, essentially because they rely on the presence of being able to detect the presence of a monoisotopic precursor peak. And as we've seen in the past, many of these monoisotopic precursor peaks don't have the same type of chromatographic traces as the fragment ions that underlie them. And then secondly, many of the times we actually don't even see a precursor peak at all. So libraries really tend to help in these types of frameworks. And so there are many different types of ways of building libraries. And I think the most common way to do this is with DDA. So one strategy for doing that is by taking a DDA experiment or a sample set that you're interested in and adding into it retention time standards. These would be synthetic peptides. They could be, in this case, actual IRT peptides from Biognosys, but they could also be just sort of any type of peptide that you have some sort of a reference for. So for example, the Pierce standard retention time standard is a really good, inexpensive standard that I highly recommend. But you could buy peptides of your own. You could use BSA, digested BSA. You could even actually use peptides that are endogenous in your sample as retention time anchors, as long as you or someone else has been able to map them onto a single line. So the way in which these strategies work is they spike in these specific peptides, in this case in blue, look for them specifically, and then take all of the peptides that they've identified in that DDA run that you don't necessarily know an index retention time for, and then essentially map them on that line based on their actual retention time in this one sample. So this derives what are called index retention times, or IRTs, for each of these different precursors. They're estimates for their retention times, but they're not necessarily, they're a reasonable estimate for what they should be. And the other aspect of this is they can be on a fairly arbitrary scale. So the IRT scale, for example, descends into negative space, negative time, and oftentimes has much larger values than the actual retention times in minutes in your actual experiment. And so this strategy actually works fairly well if you're doing back-to-back DDA versus DIA runs for building libraries, but it's not very deep, and it also involves a lot of extra work. Another strategy, and so in this case, you would actually take these same IRT peptides and then use your retention times from your first DDA run to actually map onto another sort of DIA run or DDA run. So another strategy actually is to take these types of measurements that you were getting for these IRTs and actually now that you've known something about these peptides that you've already mapped in a single DDA run, actually try to use those as new types of anchors in the future. And so this is actually a strategy that I was working on with Mac, and we did some of that work that he showed for doing DIA analysis using a DDA library where everything was put onto the same sort of index retention time using an arbitrary retention time rather than a true IRT standard. But the idea here is you would take known peptides that are common to a lot of different samples that are just the equivalent of the BSAs and the actins and the keratins that are frequently found in all of your samples, use those as new IRT samples, and then map them to a series of additional other runs that you could collect in your lab. And so this is a way for developing these sort of mega libraries across many different samples. So for example, Rob Lawrence and I published this paper a couple of years ago where we actually combined over 1,000 different runs, some internal, some external, to develop an entire phospho DDA library that would help us do DIA analysis and PRM analysis in the future. So these are fantastic methods. They do require either stable isotope labeled peptides or using an endogenous peptides inside of your system, but they're a great way to build DDA-based libraries. That said, DDA-based libraries are not without any challenges. So one issue is that they do require tons of different runs to be able to generate a clear library set. So for example, in all of these cases that we've looked at sample-specific libraries, they really require tens to hundreds of different runs in an experiment. Many times when we try to do these types of experiments, we'll have to look at 40 or 50 different DDA runs just to generate the library itself. And then if you're trying to build a pan-organismal library, you really need hundreds to thousands of runs. The other aspect is that you really also need to use lots of different fractionation methods. In this case, SCX and high pH reverse phase actually provide very different overlaps of types of peptides. Another aspect to this is that different measurements that you would get for fragment ion intensities in DIA actually are somewhat different than with DDA. And the rationale behind this, or the reason behind this, is actually due to different collision energies or different variances in how collision energy is set up for DIA versus DDA. And then finally, the retention times that you get when you've done these types of fractionation methods actually indicate different shifts in patterns and in time because of the fractionation step itself. So in an experiment where you're measuring in an actual matrix, the peptide of interest is actually being monitored in the background of tons of other peptides in general, but when you've done fractionation, you're actually only monitoring that peptide in a background of a handful of peptides. As a result, the retention times for these peptides actually shift and wobble around a bit, as much as 2 to 5 minutes on a 90-minute or 100-minute gradient. So sometimes fairly extensive. So Nick talked a little bit about trying to develop these predicted libraries, and this is actually a really excellent new strategy, which has been changing a lot of the ways in which we've been analyzing DIA experiments in just the year that this work has been out. So with a tool like PROSIT, you would end up entering in or giving in a peptide sequence. It would feed that into a neural network and then estimate what it thought fragmentation patterns and retention times would be for that specific peptide. And then these can be used to do library searching in the future. So it essentially forgoes the DDA generation step. I think this is an extremely powerful method. That said, if you search these libraries directly, they still will underperform a little bit. So for example, here I'm showing the number of peptides detected in a yeast run on the left and HeLa on the right of a PECAN search, which again, as I said, tends to underperform versus one of these SCX and high pH reverse phase DDA libraries, a combined DDA library, versus just searching a straight predicted library. And the predicted libraries do significantly better than not using a library at all, but they do underperform a little bit versus the DDA libraries. That said, they don't require any DDA experiments. And so you skip the fact that you have to generate this SCX and high pH reverse phase using 16 different injections for the yeast library and 39 injections for this HeLa library. So all of that work is essentially forgone by just using these predicted libraries. So they actually are very powerful, but they're not as powerful as some of these DDA libraries that are built on a sample-specific basis. The reason behind this is actually because of the number of peptides you end up having to search. So here I'm showing you the number of peptides in this HeLa spectrum library is just north of 100,000. It's, I think, 150,000 different peptides sequences. That's significantly less than the number of peptides in the human FASTA database or with different PTMs. And because of that, your multiple hypothesis correction analysis needs to be much more stringent when you're searching all peptides in a human FASTA. And scales as you increase with number of PTMs. This is one of the reasons why we recommend using as curated a database as possible. So for example, UniProt, SwissProt, if possible. And that using this type of strategy, we highly recommend not considering PTMs like oxidation as you would normally do for DDA analysis. Or semi-triptychs or unusually high charge states or +1s. In these cases, really searching fewer peptides actually does typically provide you with more detections and better search results. So earlier in this year, we actually published with Lindsay Pino as the lead author on this work. Seth and I, as co-authors, ended up publishing a strategy for developing DIA-only libraries and easily reproducing them, making them very easy to manufacture. And so the idea here is you would take from all of your quantitative samples, so your orange and your blue samples, we would combine them into a pool. And then in this pool, we would acquire multiple gas phase fractionation experiments, in this case 6, just like how Mac was doing for the ocean proteomics, and use these to build a library from, doing one of these FASTA searches or using a synthetic library, and then searching your single injection DIA against this gas phase fractionated DIA library. So this strategy develops in-house libraries on your samples, but instead of using 39 SCX and high pH reverse phase, highly difficult to or intensive to acquire experiments, versus just doing 6 gas phase DIA experiments, which are much easier because all you're doing is tuning your instrument to acquire the gas phase fractionation methods. So what is gas phase fractionation? It's essentially just a normal DIA experiment, except it's broken into multiple acquisitions. So for example, if you were acquiring from 400 to 1,000 in a normal DIA experiment, gas phase fractionation only means that you're acquiring in one experiment from 400 to 700, and then a second experiment from 700 to 1,000. This is a strategy that was pioneered with DIA in about 10 years ago in the Goodlet lab, and has been nicely shown in this Sonia Ting paper from 2017 as well for modern methods. For library generation, we typically break this up into not just 2 fractions, but actually 6 fractions. So for example, in this case, we'll monitor from 400 to 500 and 4 MZ wide windows. And after deconvolution, this essentially produces 2 MZ wide DDA quality or PRM quality libraries, except they're based off of DIA methods. And so if you acquire 6 of these, you wouldn't want to do this for every experiment, but it can be really beneficial for doing library generation. So the strategy with this is you would take a FASTA database and you would generate a predicted library using a PROSIT tool like the one that Nick provided. And then you would take your 2 MZ wide isolation gas phase fractionated DIA library sample sets, and then you would search these where it's very easy to detect these peptides because they're very easy to see in these experiments. So in this case, we have now situations where we've mapped a predicted spectrum or predicted peptide to a gas phase representation of that peptide. And we can combine all these DIA spectra into a new, what we call empirically corrected chromatogram library or DIA-only library. And this can be a nice advantage when you're searching single injection DIA, where you would actually be monitoring this single injection experiment or a single injection experiment set and being able to map peptides that you identified in your library to these single injection experiments. And since everything is acquired on your instrumentation at exactly the same time using DIA methods, it's very easy to line those spectra up and to be able to identify them. So the retention times are extremely accurate now, accurate to about 20 seconds on a 100-minute gradient versus accurate to 5 minutes on a 100-minute gradient. So this becomes a way to use gas phase fractionated DIA methods to refine a predicted library and make it more useful and tuned specifically for your instrument. So this strategy actually works really well no matter what source of library that you use. So even if you use PECAN to build a library to just search a gas phase fractionated data set, you can actually use that new library to detect a lot more peptides than you would typically do. Same thing for the SEX and high pH reverse phase DDA libraries, and same thing for the predicted libraries, that you get a significant boost in the number of detections that you can make. But I think the most important thing is that no matter what the source is, whether you're using building this off of a sample-specific SEX and high pH reverse phase DDA library, or you're building it off of a predicted library, you essentially get about the same results, indicating that they both sort of saturate and provide the top or the highest level of library quality. In this case, being able to do this from just the empirically predicted empirically corrected predicted library actually only uses 6 DIA runs. So this is a really powerful method for generating libraries with very little effort involved. So how do you build these libraries? I think in Lindsay's paper, this I think is really well spelled out, but I think one of the important messages from this is that you need to monitor when you acquire your gas phase fractionated runs, and this happens because retention times shift over the number of injections on a column until they settle out after about 6 to 10 different injections. And so if you see on this retention time axis on the left, you can see at about 10 injections, you start to see a settling out of the actual retention times. We really need all of the gas phase fractionated samples to have the same retention time axis. And the reason because of this is because they share very few of these peptides. And the aspect here is that multiple or these gas phase fractions typically don't share very many peptides. They only share peptides that are of different charge states. So for example, if a peptide is a +2 in the 600 to 700 range, then it will also be present in the 400 to 467 range as a +3, for example. And you can use this potentially to retention time align between all of these samples, but it's very expensive and very complicated to build this sort of spanning tree to retention time align between all of the gas phase fractions. So we highly recommend just waiting until your column settles out and then collecting them after about 10 runs or so. So given this, where we would recommend starting from is that if you need a really quick answer or you've got a fairly simple matrix, we recommend actually just, DDA actually does really well with this using the normal scaffold pipeline. We think that this is still a really good way to analyze many types of samples if you're not trying to do a lot of quantitation, or you're just trying to get a simple answer for what's in the sample, or you're analyzing gel spots or something that, or gel bands where you have a simplified matrix. DDA performs really well in these circumstances. And so we still highly recommend considering to use it. That said, if you're trying to do a larger quantitative experiment, we find that DIA actually tends to produce better quantitative data in the long term across lots of different samples. If you're looking at less than 6 samples, we recommend this strategy that I believe Sue may talk about in a bit, where you actually use multiple gas phase fractions for single samples, individual samples. So if you've got less than 6 samples, this 2 times gas phase fractionated DIA library, DIA analysis method is actually a fairly good strategy. And here you would just search that with a predicted library. If you have more than 6 samples in your experiment, we recommend this idea about building a gas phase fractionated library and then searching that with one of these predicted libraries for doing that type of analysis. We find this to be a very practical way of generating a very deep library specifically for DIA on your instrument. There are circumstances where you might want to build a DDA library still, and in these cases, might be around PTMs or situations where you have peptides where that are just not very well predicted by the engines like ProZen. So with that, I'd like to just thank some people from Genome Sciences and from Proteome Software that have helped out on this work, as well as Novo and the PROZIT group at the Technical University of Munich. So with that, I'd like to close. And I'd be happy to take any questions if there are.
Awesome. Thank you, Brian. That was a great presentation on libraries. Lots to think about, and it's clearly a very important aspect of designing any DIA experiment. So something to spend a fair amount of time thinking about, and we see there's a lot of options to have.